where, so we are writing the etiology of this particular disease is still unknown or it's idiopathic in nature. But we are assuming various factors are involving in the development of that particular disease. If you talk about, primary hypertension, so we are assuming that, so 95% cases of primary hypertension only. And if we talk about what are the exact etiology of primary hypertension, we don't have an answer for it. We are assuming various factors, lifestyle disorders, stress, diet patterns, environmental factors, and so on are contributing for the development of that particular disease. So unless and until if we don't know the etiology, how we will cure the disease? We can't cure the disease. So, network pharmacologies to certain extent will help you to understand the pathogenesis of the disease as well. What are its etiologies? What are its pathogenesis? And how your molecules can be play a role in the preventions or treatment of that particular disease. So network pharmacology, what actually it is? It is devoted to understand drug pharmacological mechanism in network perspective. So, if you see this word carefully, pharmacology, every one of us knows what is pharmacology, study of drugs. So what word has been added to pharmacology? The word which is added is network. So what do you understand by network? In layman terms, if someone called, if that XYZ persons have lot of network, so that means they have lot of connections with the other target with respect to pharmacology, right? So network pharmacology is devoted to understand drugs pharmacological mechanism in a network perspective. So it's an emerging discipline useful in the drug discovery, and it combines genomic technologies. You have to be extract out the data with respect to genes, with respect to targets, and you have combining the system biology and pharmaceutical chemistry means knowledge is also required because you are dealing with the structure of the compounds. You should know means how your structure of the compound can affect that particular target. And these all things we are doing with the help of computational biological tool, right? So what is required to do the network pharmacology? If someone of you ask me, I want to do network pharmacology, I want to apply network pharmacological techniques. So what is required? You required only the laptop or computer systems with good internet connection. That's it. And whatever we are going to cover, I am going to cover only those databases which are freely available, where you can access the data by creating your login ID and password. You can use that data. So it's a new frontier discipline of systematic drug research in the era of artificial intelligence, and globally network pharmacology, we called it's regarded as a next generation's medicine research mode. So, so before going into the network pharmacologies, first, sometimes students ask me what skill is required to do the network pharmacology. The particular, I always answer to this question is, don't forget your basic knowledge. Basic knowledge is must, because these all are the computational techniques. Whatever you will add in the computers, you will get something in the form of output. If you are adding something in the cytoscape is the one of the software which are popularly used in the network pharmacology. So maybe many of you knows about it. So what you are doing, you are putting the informations in the cytoscope. It will create the network, but without the knowledge of molecular pharmacology, you can't do a network pharmacology in a good way. So how you will get the knowledge about molecular pharmacology? So you have to know the signaling pathway. Unless and until if you for example, you are working in the metabolic syndrome, and if you don't know about what are the possible signaling pathways which are involved in the metabolic syndrome, how you will do the network pharmacology in a better way? Whatever the system will give the output, you can't understand it. You can't correlate that signaling pathway. So first important thing is, whatever area of research you are working, you understand its signaling mechanism. What are the mechanisms which are involved behind that particular disease? Molecular mechanism, molecular signaling pathways, all that understandings you should have before doing the network pharmacology. So, so that's why there is a role of system biology. So you can see that system biology means you have to understand how proteins are interacting with each other, how your targets, whatever the targets, how they are interacting with each other, and particularly producing the particular effect. So you should have a good knowledge about the system biology as well. How drugs are absorbed. So every one of us knows, being in a pharmacy profession, we knows pharmacokinetic, we know pharmacodynamic as well. But somewhere we are lacking is about the intracellular signaling pathways, because these are the signaling pathways, day by day new signaling pathway has been explored, new role of earlier signaling pathways, so they are changed over the times. So you have to be keep updated yourself. If we are talking about any particular signaling pathway, if I am targeting that particular signaling pathway, how much it will be beneficial, how much it produce the adverse effects. So, pharmacology, everyone of us knows. So, I am going to skip. So, as I said, before doing the network pharmacology, please focus on basic of pharmacology. So, every one of us knows that 80%, approximately 80% of the drugs are showing their response through interacting with the receptors. And how many types of receptor? Receptor are of majorly four types, right? Enzyme kinase link receptor, ligand gated, G protein coupled, right? Nuclear receptors. So, you have to understand when drug is acting on the ligand binding domains in the extracellular domain of the receptors. So what signals, intercellular signaling pathways are ongoing? So you knows about GPCR, how GPCR act. There is a GS, GQ, GI isoform. If GQ is activated, then your phospholipases will do the degradation of PIP2 into IP3 and DAG. IP3 has a receptor on smooth endoplasmic reticulum. Smooth endoplasmic reticulum is the storehouse of the calcium ion. So more calcium will release, more calcium will interact with actin and myosin filaments and result in the contractions, right? That is so, what I mean to say, these signalings you have to be understands in a better way, how these signalings are happening, how molecules are showing their response by interacting with a particular target. So like these pictures, you can see this is about the nuclear receptor. So, in nuclear receptors, there is a heat shock protein who is masked this ligand binding domain. But when your ligand is binding, heat shock proteins will be moved. And like, steroids are showing their action through the nuclear receptor. So it will be released. So it will be, transcription and translation process is going to be happens. And there is a new synthesis of the proteins. Like you can see, this is about the enzyme kinase linked receptors. I am not going in detail because, every one of us knows about it. But what my purpose is, go through the basics before picking up, before going or, into the network pharmacological approaches. You have to understand if we are talking about enzyme kinase linked receptors, so how intracellular signaling pathways are ongoing. What is the conformation of the receptors in its inactive state when ligand is binding on the ligand binding domain? So these monomers will be converted into the dimers, right? So if you can see that your ligand is binding, so these monomers will be converted into the dimers, right? Then there is a phosphorylation of substrate, and phosphorylation of substrates will going to activate tyrosine kinases, right? And then specific amino acid residues, and you will get the response. And this is about your GPCR signalings. You all knows about it. So GS, GQ, if GS is, is activated, then your adenyl cyclases will do the degradation of ATP into CMP, and increased CMP will do the contractions, right? But again, these are our classical pharmacology means, everyone knows about these receptors. Every student knows about these receptors. And of course, we have to just go through about what are the important, these intracellular mechanism. But there are a very important signaling pathways, they have a role in each and every disease. So, whether you are working in diabetes, whether you are working in neurological disease, whatever your area is, each one of us knows about these important signaling pathways. So important signaling, one of the important signaling pathway which is involved everywhere is apoptotic signaling pathway, right? So you have to go through that apoptotic signaling pathways, how increased oxidative stress results in alteration in mitochondrial membrane potential. Then there is a, activations of your caspase independent pathway. If your caspase independent pathway is activated, there is a apoptotic inducing factors, endonuclease G. If it's activated, caspase dependent pathway, then your cytochrome C, APAF1 and caspase 8, and ultimately it will do the degradation of DNA. And DNA fragmentation is one of the hallmark of your apoptosis. So that is one of the signaling pathway. Then P53 signaling pathway for the apoptosis. P53 in mitochondria, there is a apoptotic protein and anti-apoptotic protein. Apoptotic proteins are BAD and BAX. BCL2 is your anti-apoptotic protein. So when your BAD and BAX level is increased and your BCL2 level is decreased, so it results in imbalance, and that imbalance will further activate your apoptotic signaling pathways, and that result in the activation of caspase 3. Caspase 3 will act on the DNA and do the fragmentation of the DNA. like that, these are the other pathways with respect to your apoptotic signaling pathway. So, but what I mean to say, uh, go through these apoptotic signaling pathway, because apoptosis is, normal physiological cell death. So every one of us knows, if you are going to apply network pharmacological techniques, so you should understand about the signaling, about the apoptotic signaling pathway. Because when you get the results by the computers, maybe you are getting some results about the caspase signaling pathway. So you have to understand, if caspase 3 is there, so obviously caspase 3 will result in the DNA fragmentations, and that is the apoptotic signaling pathway. If we are talking about, if you are getting by the network pharmacological results, you are getting information about BAD and BAX proteins, BCL2 proteins. So, BAD, BAX, BCL2, these are the proteins which are involved in the apoptosis. BCL2 is inhibiting the apoptosis. BAX and BAD are the inducer of the apoptosis. So, when you understand these signaling pathways, so you will understand the results in a better way. Like another signaling pathway which every one of us knows is, should know before doing the network pharmacological technique, is immune signaling pathway. So immune system is another system which is involved everywhere, right? In every part of the bodies, there is a immune cells playing a role, major role. So you have to understand how immune signaling pathways are activated. What are the toll-like receptors? I hope you know toll-like receptor and nod-like receptors. So whenever any antigen enter in our bodies, these are sensed by some specialized proteins which are present on the WBC. So these proteins we call toll-like receptor. Toll-like receptor, TLR, these are the sensor. So what they are sensing? They are sensing that whatever the things enter in our body is non-self. It's exogenous substance. It's not body part. So your toll-like receptor should have the ability to differentiate between self and non-self. Once it sense it, it's a non-self. So it's going to activations of TLR dependent signaling pathway and TLR independent signaling pathway. And there are a number of TLR. Now there are TLR 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11. So every now scientists have identified that which TLR is responsible for the sensing of which protein. If there is a viral protein, so particular TLR3 will sense it. TLR4 will sense viral protein. This particular TLR will sense this particular proteins. You know. So with the advancement, we are getting to know much more information, and these are the sensors who will sense it. And after sensing, they are going to activate various signaling pathways like MAP kinase. You might heard about MAP kinase signaling pathway, NF kappa B. All these pathway result in the activation of nucleus, and it results in the transcription and translation process. And during that process, a lot of cytokine will release. And the purpose of the cytokine is to kill that exogenous substances. So when your innate immune, that is a part of your innate immune system, right? So when your innate immune system is unable to handle, unable to kill that exogenous substances, so what happen next? So next thing is your that particular antigens or exogenous substances will be presented to the adaptive immune system. And adaptive immune system, there is a T cells and B cells, right? So antigen presenting cells will catch that pathogens and present to the adaptive immune system, particularly T cells. And this presentation mechanism is also specialized. There is a MHC molecule, major histocompatibility complex. If your antigen is catched by MHC2, then your antigen will present to helper T cells. If it catched by, MHC1, it will be presented to cytotoxic T cells. And this is a, very comprehensive presentation mechanism. And once your adaptive immune system will sense it, then it will result in the activations of your helper T cells, cytotoxic T cells that will goes under the process of clonal expansions, right? And these release cytokines will simultaneously activate your B cells into the plasma cells, and plasma cells will produce antibody against it. So what I mean to say, every one of us should clear about the immune system signaling pathways. So, whenever you got the network pharmacological results, you can get information about the immune signaling pathways as well. So that time you can correlate it. Okay, I am getting this immune signaling. I am getting this MSC signaling molecules. I am getting this, the helper T cells, or particularly this particular signaling pathways. So what is its role and how it plays a role in particular my research work, right? So, there are many pathways. I am not going in details. Maybe it will become little bit boring. I will enter into the network pharmacology and how to, if anyone of you would like to use these approaches, how to use it. But major problem what I feel is uh, various now so researchers are using these network pharmacological techniques, but without understanding about the signaling pathway. Please understand the signaling pathway in much more detail. Then so your network pharmacology results will become very much interesting to you. Computers are just to help you, so but whatever the informations how to understand these signaling pathways, you have to first understand about it. This is the so TLR signaling pathway. So you can see that these signaling pathway are very complex to each other, because they are not playing role in the one particular activities. They are helpful in the cell survival, cell divisions. There are so many roles are there. So that's why when you are discovering a molecules or coming up with the molecules, you are saying that I have discovered this molecule and it's going to inhibit this particular target. So you have to understand what is the role of that targets in normal physiological conditions. If you are targeting that targets, so what will happen if you are inhibiting it? What is the effect on normal physiological function of the body? Because so whatever you are targeting, that also playing a role in normal physiological functions, right? So this is the just a presentations of how this is the dendritic cells which act as a antigen presenting cell, how it is presenting the cells to the helper T cell, how it is presenting to the cytotoxic T cell. So as I said, there is a specialized mechanism. These are the co-stimulatory molecules, and once it sends, it goes under the process of clonal expansions, and they will expand their numbers, right? And release a lot of chemicals which will help to kill the pathogens. If it's a viral pathogens, then interferon will release. Interferons are antiviral in nature, right? So depending upon the nature of pathogens. Then another signaling pathways, you might heard about JAK-STAT signaling pathway. Lot of research is ongoing on JAK-STAT signaling pathways. Some molecules are under clinical trials as well. So you have to understand JAK-STAT signaling pathway as well. So JAK is Janus kinases and STAT is signal transducers activator of transcriptions. So there are two things. JAK is Janus kinases. STAT stands for signal transducers activator of transcriptions. So these particular pathways, if you look for it, you will find that these are the enzyme kinase linked receptors. Whatever we have discussed earlier, they are monomer. Then whenever ligand binds, so ligand in this case will be growth factors, can be your interleukins. It will bind in the extracellular domain of the receptors. Then it will be going to activate these JAK-STAT signaling pathways. To read about JAK-STAT signaling pathway, as I said, there are a lot of research is ongoing where people are identifying STAT inhibitors, where people are identifying JAK inhibitors. So we have a drugs on Janus kinase inhibitors as well. These kinases are playing a role in normal in number of processes, right? So, but you have to be careful about their adverse effects as well. Got it? So there is a stat 1, stat 2, stat 3, like that we have a number of molecules. I am not going in details, but of course, there is another pathway which is popular is, so that is the PI3 AKT signaling pathway. PI3 AKT signaling pathway, if you see it, it also belongs to your enzyme kinase linked receptors, where your ligand is binding. And after that, there is a dimerizations, and in dimerization, there is a phosphorylations of the substrates, and PIP2 will convert into PIP3. Then it will result in activation of AKT. AKT will further activate mTOR. mTOR is mammalian target of rapamycins. So you might heard about PI3K, AKT, mTOR signaling pathways. And they have a role in number of activities. So now research is ongoing. We have a mTOR inhibitors drug as well. Particularly, if I remember correctly, it's for epilepsy. Rapamycin is a mTOR inhibitor drugs, right? So if you are working, you should know about these mTOR signaling pathway, PI3 AKT signaling pathway, and you can see that there are a PI3 AK inhibitors, people's, and we have a drugs AKT inhibitors, right? GSK3 beta is another important signaling molecules where a lot of research is ongoing, right? So these are the, how these, what actually ongoing from 1971 to 2023, what research is ongoing on these particular targets. So you can see that how many molecules has been identified, mTOR inhibitor, viral infection in old age patients, immunotherapy with mTOR inhibitors. We have studied immunotherapy as well. Rapamycin enhance the potential of anti-tumor vaccines as well. A lot of research is ongoing with by understanding the signaling pathway. So you can see that these are the phase one. This blue one is phase one clinical trial, green one is phase two, and these are the phase three clinical trials. So now, I hope that much background is enough. I hope I convey my message is please go through the signaling pathways, and these are the signaling pathway which are, so every one of us need to understand in detail about the signaling pathway. If there is a phosphorylation, so after phosphorylation, what's happening, how it's going to activate, what is its role in the normal physiological functions, and what is its role in the pathogenesis of the disease as well. So once we understands about our pathogenesis of the disease, where we want to apply the network pharmacology, so first we have to understand where we can use these network pharmacological approaches. So importance of network pharmacological approaches is you can identify the potential drug candidate. If you are so working on new chemical, you are from pharmaceutical chemistry background, you have synthesized a new chemical entities in your lab, you have done the characterizations, NMR and all other techniques, you have characterized this is the molecule which you have synthesized. So after that, before doing the in vitro testing or in vivo, you can apply the network pharmacological techniques to get an ideas, how this molecule is going to be act by activating which signaling pathways in that particular disease. Of course, when you are synthesizing a molecules, you are normally synthesizing by keeping so disease in your mind. Okay, you are synthesizing the molecule for the diabetes, for Alzheimer, and so on. You have designed analogs and you have done that part. So after that, before doing the experimental studies, you can apply the network pharmacological studies to understand what are the possible mechanism by which your molecule can show the actions, that particular section action in that particular disease. That is a one. If you want to understand drug mechanisms, right? If you want to understand drug-drug interaction as well, you want to, for example, you are from PharmD background, or from pharmacy practice background, where you want to understand, this is the drug-drug interaction. You are getting the reports from the post-marketing surveillance. You got to know that these two drugs could result in, for example, for example, hepatotoxicity. So now you want to understand how these drugs, what is the possible mechanism, how these drugs could result in hepatotoxicity. You can apply the network pharmacological techniques to understand that part as well. And you can identify the possible mechanism of adverse drug reactions as well. There are many adverse drug reactions which are happening, and we don't know about their possible mechanism. So if you want to identify what is the possible mechanism of that adverse drug reactions, you can apply the network pharmacological techniques. So in our lab, we are applying network pharmacological techniques to understand the mechanism of the drugs, and also to identify the possible mechanism of adverse drug reaction. And particularly, we are first identifying the novel signal of the drugs by using, VG-based and FAERS data. We are applying some statistics as per regulatory guidelines. And when we are identifying a novel signals, so it's a novel signal, so obviously its mechanism is unknown. So to identify its potential mechanism, we are applying the network pharmacological techniques. So popularity of network pharmacological techniques, if you just type in a PubMed word network pharmacology, you will find that year by year, how many studies are being published by using the network pharmacological techniques. So you will find that these are the articles in the PubMed, and you will find that these number of studies are increasing year by year. You can get little bit dip here, but dip is due to because it's not complete data of that year. It's the mid of the year. So that's why there is a little bit decrease. But when you consider the complete year data, it will be increased. So what this statistic shows? It shows that these network pharmacological techniques are, year by years, researchers are using it for their research purposes. And now, if you do little bit more analysis about the article which you are getting on the network pharmacology, you will find that most of the articles who are using these network pharmacological techniques are Chinese researchers. Now question arises, why Chinese? Why Chinese are using these network pharmacological techniques? So if you go through little bit detail, you will find that there is a TCM. Many of you might heard about TCM. TCM is a traditional Chinese medicines, right? So traditional Chinese medicines, in China, they are doing the treatment of the disease by using the traditional Chinese medicines, right? So traditional Chinese medicine is just like a herbal medicines. If you see the composition of traditional Chinese medicines, you will find that they are using particularly the herbs. So if you go through these articles, you will find that why they are using these network pharmacological techniques for their traditional Chinese medicines. So you will find that a lot of articles in the literature where they have used network pharmacological studies to understand the mechanism of traditional Chinese medicines. So these these techniques are helping them to generate the scientific evidence. What is the problem with the herbs? What is the problem with the herbal medicine? The problem with the herbal medicine is, if so if you are talking about the herbal medicine at the international platform, you are saying that no, we are using this particular herbal since long times and it's very effective. No one will believe on it on in international platform. They will ask you, show me the data, show me some scientific evidence. So, network pharmacology will help to I will not say it's completely help, but it help to a certain extent to generate scientific evidence for your medicines. Particularly with respect to, so if you go through these Chinese paper, you will find that they are exploring, they are using the network pharmacology for their traditional Chinese medicines. And you will find that they are, then after that, they are proving it by the experimental studies as well. So it will generate the scientific evidence. Now, next question should be arise in everyone's mind is, why not India? Why not we are, if you go through the how many studies with respect to network pharmacology has been published with respect to network pharmacology last 10 years, you will find number is very less. Why? And what is the means and many times many institutes are telling that they don't have these facilities, so that's why they are not able to do the research work and so on. What to do the network pharmacology, as I said earlier as well, what is required? You need only the computer system. I think every institute have a computer system, everyone has a laptop. And of course, if you are asking these databases are commercial one, of course there is a commercial databases as well, but we have a freely database as well. And researcher have done a lot of publication by using the data of these databases which are freely available in open platform. So now, so that's why question obvious: why not India? India has a rich, traditions of herbal medicines, Ayurveda, Unani, Siddha, and so on. So why we are not using these network pharmacological techniques? We have a, if you see the list of top 2% scientists, you will find that a lot of Indians are there. So we are not behind any countries. Why? And the Indians are particularly good in the programming. Indians are very good in the computers things. So that's why if you see that in US as well, you will find that a lot of Indians are doing that work with respect to computers. US minds are not that much, they are not understanding that better way as that Indians can understand about the things. And of course, infrastructure. So, so every institute of India has this infrastructure. So question is, why we are not using it? Maybe we don't knows how to do it. What are the steps? Maybe we means we can explore these techniques and we can generate a scientific evidence for our herbal molecules. But this is a one of the area where you are applying the network pharmacology. If I am not asking you to start working on the herbals, if you are not interested for herbal or your research area is something else, you can use these network pharmacological techniques for your areas as well. But with respect to herbals, why not? Why we are not using it? So to understand, so that's why understanding is important. And by conducting such kind of FDP, so we will get to know that how to do this, what are the challenges, how to do the interpretations, and we can do many things. And beauty of these things is you are not dependent on anything. You have to be very good understandings about signaling pathways, and if you know the steps, you can do it. So, understanding for understandings, I am going to start with simple network. So, simple network, you will find like this. This is just a simple network. There is a circles, and these circles are connected with the lines. This is the A, B, C. So, circles in terms of network pharmacology, these circles we call nodes. So, in network pharmacology, if someone is talking about node, you have to understand that are talking about these particular circles. And these circles can be your gene. What these circles represent? These circles can represent your gene, proteins, metabolites. It depends upon what kind of network you are doing. If you are doing gene network, if you are doing protein, if you are doing metabolites network. So it depends upon that. So circle represent the node, and the line which connecting these circles that we call edges. And what it shows, these lines are showing the relationship between these two circles. So if it is very close to this one, so you will conclude that this have a strong relationship with this particular node, right? So circles are the node, and the lines which are connecting the circles we call edges. So if someone is talking about node and edges in the network pharmacology, so you have to understand this. So if you see the history, how this network pharmacology have been developed, so it's a very long back, 18th century, by the Swiss mathematicians. So they have eventually solution to the famous this bridge problem. So you just it's not possible to explain each and everything, but you just go through this bridge problem, what actually this bridge problem is, and how these scientist mathematician has resolved that bridge problem. So the concept of network pharmacology started from there. So the German city of Königsberg, present day, say Klingard, is situated on pre-Golia rivers. Geographical layout is composed of four main bodies of the land connected by a total of 7 bridges. The question posed to the Euler was straightforward. The questions to that mathematician was it was possible to take walk through the town in such a way as to cross over every bridge once and only once. And that is known as, later it's known as Euler walk, because this is the with the name of the scientist. So it depends upon the graph theory. So graph theory is ultimately help in the study the relationship. So network pharmacology is depend upon the graph theory, right? So with such kind of network, you will find that there are a undirected network, directed graph. So what are these? What are the differences between these? Very simple differences you can see in this picture. This one is undirected graph. This one is directed graph. So undirected graph are those graph where no direction is given. It can be gone in either way, 1 to 2 or 2 to 1, right? But in directed graph, you can see there is a directions, 1 to 2, then 2 to 4, then 4 to 3, then 1 to 3. So there is a directions. So that we call directed graph. So, if someone of you is working in the network pharmacology, you will find that when you are uploading your network in the cytoscape, you know, in cytoscape, it's asking about directed or undirected graph. Got it? So what is directed and what is undirected? So that is the meaning of directed, undirected. There is another graph you will find in the network pharmacology is weighted versus unweighted graph. So weighted, as its name indicate, you are giving some weightage, right? So weightage to the path. So you are giving weightage from B to C 1, then 2, then 10. You are giving some weightage to the graphs. So that kind of graphs we call weighted graph. And where you are not giving the weightage, that we call unweighted graph. Then there is a acyclic or cyclic graph. So you can see that acyclic can be directed, acyclic can be undirected. Like that, cyclic can be directed, cyclic can be undirected. So cyclic, acyclic directed means it's a acyclic graph. Acyclic means it's not a cycle. So you are going from A to B, then B to D, but now D to C you are not going. There is a direction is not from D to C. So it's not completing the cycle. That is the acyclic graph, such kind of graph. So then directed acyclic graph is where you are giving the directions. Here is a undirected acyclic graph. Here cycle is not. You B to E, then D to C, but you are not returning back to V, right? But in cyclic directed graph, you can see that B to D, D to C, C to A, and you are returning back to B. And even though if it's undirected graph, but if it's cyclic graph, you can return back, so if you are your direction is not given, but it's a cycle. I hope every one of you are getting this, right? So acyclic versus cyclic graph. Then there is a sparse versus dense. Sparse, you can see that that is not that much dense connections between the edges. But when you see that these nodes are connecting with a lot of edges are there, that is we call dense. So important terms related to network is a well computed mathematical representations of various connected node and edges. So there is a terms with respect to network is hub. So what is hub with respect to network pharmacology? Hub is a node with a higher degree. So now question is what is degree? Degree is the number of edges connected to a node. Then what is betweenness? Betweenness is the number of shortest path that go through a given specific node. And what is shortest path? Shortest path is the minimum path between any two nodes in a network. That is we call shortest path. So you can see in this picture clearly what actually the hub, what actually the degree, what actually the shortest path, right? So you can see this one is the hub, a node with the high degree. And degree is the number of edges connected to a node, right? Betweenness, the number of shortest path that go through a given node. So these kind of terms, so when you are doing the analysis of network, so in that case, so you have to write about hub, about degree, betweenness, shortest path. So as I said, there are different types of network: gene regulation network, protein network, signal transduction network, metabolic network. It depends upon your area of interest. Now, important part is what are the common databases and tool which we are using in the network pharmacology, and what are the steps, right? So first step is identifications of targets of the compound. So now, for examples, you are going to apply network pharmacology, for example, for the herbal molecules. And so if you are working on a particular plant or extract, so extract might contain number of phytoconstituents. One extract can contain 50 or 100 phytoconstituents. So what if you go through a studies which are published in the literature, you will find that what some researchers are doing who are working in the herbals, they are first preparing the extract of particular plants, particular part, and then they are doing GCMS or any other analytical technique to identify particular phytoconstituents in that extract. And after that, they are applying the network pharmacology. And this become a experimental plus network pharmacology part. You know, uh, sometimes, what problems you can face with respect to network pharmacology is, for example, you have done network pharmacology. By the network pharmacology, you have identified particular protein targets that my molecules is can show the action by modulating these targets. So, sometime reviewer will ask you, okay, then show me modulation of these targets by the experimental approaches. Are you getting it? So, sometimes, so these antibodies for the Western blot, these are costly one. So, due to that limitations, if you are unable to do the experimental part, so what you can do is you can do first, so you can prepare the extract, you can do the animal activities, right? And then in the extract, you can identify what are the major phytoconstituents in that one. And you can apply the network pharmacology with that major phytoconstituent which you have identified by the analytical techniques. That is the one way to do that, to resolve that problem. Another way to resolve the problem is, or to resolve this question is, uh, you can first do the experimental studies, animal experimentation studies or in vitro studies, and you show that this molecule is effective. This molecule is showing the response in that particular disease, and then we have applied the network pharmacology, and we have identified that this mechanism, whatever we are getting is, whatever the effect we are getting is, this is the possible mechanism behind it. And then you last part, you can write that further these can be experimentally explored. I hope it's clear, because so you will face these questions when you are first applying the network pharmacology, and then you are doing the experimental studies. And in experimental, if you are not connecting the network pharmacological finding with the experimental, so question will be arise and you can't defend it in a proper way. How you will defend it? By network, you are showing that my molecule is going to modulate MAP kinase or NF kappa B pathway, but in experiment you have not done the MAP kinase and NF kappa B. You have just checked the activity of the compounds in the animals, so you can't defend it. So that is the way how to deal with this such kind of problems, right? So first step to do the network pharmacology is identifications of targets of the compounds. So as I said, if you are working on herbals, or if you have a pure compound, or if you are working on a drug repurposing, so in that case you have a molecules. So you have to draw the structure of the compounds. So first step is you have to identify the targets of the compounds. So from where you will get that information? So you have a lot of web server. So web servers, what are these web server? Web server are, you can see the name. Swiss target predictor, C database, Stitch database, drug binding database. Up to this, these are the web servers, which will help you to find out the possible targets of your compounds. So now question might be arise in your mind, if we are identifying the targets of the compound, why there is a need to search all these databases for the same work? The answer to this question is we have to search in all these web servers. And if you are getting any other additional server apart from these, go for that as well. The reason is because we don't want to miss any target. And particularly, we are using the databases or these web servers which are freely available. So freely available databases, most of the times, these are not updated one. So, for example, Swiss target predictor is updated in 2020. So last 5 years, there is no updation is done. So in last 5 years, there may be a new targets has been identified. So you don't want to miss any targets. So what comprehensive approach is, you have to use all the available web servers. So first step to do the network pharmacology is identify the targets of the compounds. That is the first step. Second step is you have to be identify the targets which are involved in that particular disease. And that depends upon your area of research. As I said, if you are working on diabetes, so you have to be pull out all the targets which are involved in the diabetes. So for that one also you have a web servers that are the DigiNet, GeneCard, OMIM, HPO, Malacard. So from these you have to be pull out the Excel files. That is the second step. And these two steps are the most important steps for the network pharmacology, because whole network is depend upon these two steps. If you go through the literature, you will find that people have used only the DigiNet, but that is not the right way. You have to use other one as well to avoid the miss out of any targets. And even though we are doing the data mining of literature as well. So next step is, once your Excel files has been prepared, so obviously, so what happens when you are doing same thing in the different web server, there might be a duplicate. So by using the Excels, you have to identify the duplicate and remove the that duplicates, and then you make the one comprehensive file, because you have a one Excel file by the Swiss target predictor, another Excel file by the C database, another Excel file. So you have a 3, 4 Excel files, right? And that Excel file is containing the targets which are in which your compound can hit. So and there is a possibilities you will have a various duplicate as well, right? So that duplicates you have to identify in a Excel. There is a options where you can click and you can identify the duplicates and remove it, and then your comprehensive file will be prepared. That is the first step. Like similar way, you have to be do the disease. Then the third step is, now you want to see which targets are the common one, which are overlapping with the compounds and overlapping with the disease. That is your purpose, right? So for that one, earlier we are doing by using the VLOOKUP formulas in the Excel, but now we have a Vani tool. In Vani tool, in one side you add the target which your compound can hit, and another side you add the target which are involved in the disease. You will get the common target. The next step is, now you want to, because these targets are not in isolated form in the body, they are interacting with each other. So you have to check the interactions among these common targets. So for that one, you have a tool like string, biogrid, dip, stitch, inect. But now for these steps, you can use either one tool, because here there is no point to use all these tools for the interaction among the target. You can go with the string or biogrid or dip or stitch or inect, right? Then next step is Cytoscape. You can install it in your laptop, and Cytoscape is a tool where you can create the network, where you can visualize, where you can analyze the network, and then you have to do the SR plot, which will help you to find out the pathways which are involved in or enrichment. We called it. So because up to that you are getting only the targets, because these targets are making some pathways, right? So in one pathway, there can be multiple targets, right? So these are the steps, right? So first step is, as I said, identifications of the compounds, targets, right? Any question up to this? Okay. So identifications of the compounds or targets, first step. And as I said, to this is a very simple means very important steps. I can't say simple, but these tools, whatever the web server I am telling you, these are very easy to use. the use of the web server is not hard to use, so what is required in Swiss target predictors, but before using these web servers, please go through the detail about the web server. What is this web server, how it has been created, what kind of data it has, then only you can use this web server in an effective way. Otherwise you can't understand. If you don't understand what is FP2 in the Swiss target predictor, so when you are writing your research studies, you will face a problems, right? And reviewer will ask you, you have not added in a much more detail about this. So the always it's recommended, whatever the web server you are using, please go through it. Please understands what actually the database is. But many students are not going through these databases detail, and they are facing the problem when there is a viva or any questions. They will ask you, if they you have mentioned that I have used this target predictor, they will ask you, okay, then tell me about FP2 value. Tell me about 2D and 3D conformers. Tell me about cutoff values. So, if you don't have an answer, so that means you have just go through the web server and you just added the structure of the compound and got the result. So that is not the right way. So you have to understand these ES52 values and threshold values. By default, it's set 0.85. But if you are changing it, this threshold value, what will happen? Below threshold value, the known activities are not similar enough to be displayed. So sometimes we are changing this threshold as well. But again, it depends upon your projects, right? So that what I mean to say, you have to read about, and each and every information is available. Whatever the web server you will find, there is a research article behind it. So in that research article, you will find that how this web server has been created by the researcher. And again, to use this web server, anyone can use it. So even the person who is not from pharmacy background, they can also use it. It's that much simple. But the informations or the knowledge about the web servers, that only the persons who are working in the field, they can understand it much better. So you have to be select the species here if you are depending upon the your experiments, if you are looking for. So you have to select the species, and you have to understand that Swiss target predictors, for example, you are planning to do the studies in the rabbit. So rabbit species data is not there. So what is the purpose to use the Swiss target predictor if you are designing experiment in the rabbit? So what I mean to say, you have to be understands about the web server first, then use it. And again, to use it, what you have to do? You can either draw the structure of the compound here, or you can get put the smiley notation of the compounds here and click on predict target, just it, and you will get the results. And you can export the results. Here is the CSV Excel file is there. You click on the Excel file, you will get the informations in the Excel file, right? And this is the informations which is telling like this you will get. But these are the targets which your compound can hit. These are the common name of the target. These are the uniprot ID. These are the Campbell. These are the targets. These are the probability. So you have to understand what this probability is showing. If someone is talking about 0.346, what actually it is. So sometimes we have to keep on the cutoff values, where you have a lot of targets are there and your network is becoming very complex. So in that cases, you have to put the cutoff. So to put the cutoff, you should have a certain criteria. So what are these criteria? So that you have to look for the p values, so 3D by 2D activities as well. Then another one is C database. C database is based on. I hope I am on time, or I have 10-15 minutes more. Hello.
Yes, sir. You can proceed.
Okay. So C database is again free of charge, but in C database, this is provided by Department of Pharmaceutical Chemistry at the University of California. But in that C database, when you go through the C database, you will find that they are reflecting one message. Message is you can use this database at your own risk. So these databases, because these are freely available, so you all knows data is if you are submitting any molecules which is not patented and you want to patent it, so the thumb rule is first do the patent, then you use these kind of web server. Sir, and sir, these web server who has created that, they are also sir, flashing the message that you can use this web server at your own risk. We are not responsible if someone has copied your molecules, right? So if there is any confidentiality project, so don't use these kind of web server. And again, it's very easy to use C database. You just This is a screenshot of the web page of the C. Just type in a Google C. What do you have to do? You have to put the smiley notation of the compound and click on try C. Then like that, there is another one is Stitch. And Stitch is, again, database. It's freely and commercially available. So you can use free version of the Stitch. Again, you have to add the smiley notation of the compound, or you can upload the file from the CamDraw or any other tool and click on the search. There is a drug binding database. This is, database created by School of Pharmacy and Pharmaceutical Sciences at University of California. So it's again freely available. So you can use this drug binding databases where you can add the name of the compounds and click on go. You can get the Excel files. Then there is another one is CTD as well. In CTD also you can get the information about the target which your compound can hit. So you can get the information like this, Then step two is identification of disease target. As I said, DigiNet is the most popular one. DigiNet Plus is the, for commercial purposes. So in DigiNet, you have to create your login IDs and passwords. You have to do the sign up. This is my login ID and password in the DigiNet. You add the name of the disease, click on summary of gene disease associations. You will get all these the targets which can be involved in the that particular disease. So in this case, we have identified the targets which are involved in the drug resistance epilepsy. Then there is a gene card. Gene card is another popular one, and so it's particularly you will get the informations about the disease targets which are involved in that disease. There you will get the relevance score. So in the gene card, this is the web page. You add the name of the disease, you will get the targets which are involved in that particular disease. There is a man-OMIM database is also there, and particularly if you are working on genetic disorders, hereditary trait, you can't miss out this man-OMIM database. You will get the informations from the man-OMIM database. Again, you read about, so about this database, how these, what is the basis of the informations in these databases, right? So man-OMIM is another one. Okay. So then this is the online version of OMIM was created in 1985 by the collaborations between National Library of Medicines and William H. Medical Library at John Hopkins. So, these big universities have created these databases. So their data is authenticate, you can trust on these data. But important thing is you should understand how to use it in a proper way. There is a OMIM is another popular one. And when you are working on a human gene and genetic disorders, OMIM is another important database. It's a 50 years has been completed where you can add the name of the disease and you can get the informations about the targets which are involved in that particular disease. It's NIH supported projects. Then there is a HPO. HPO is a human phenotypic ontology database. This is the web page of HPO where you can add the name of the disease. Then there is a Malacard. Malacard is another important database with respect to diseases. And this is the web page of the Malacards. The Malacard, the human disease database. So you put the name of the disease, you will get the informations about targets which are involved in that particular disease. So up to this, whatever the two steps are there, you have done. One is identification of targets which can be hit by your compound and the targets which are involved in that particular disease, right? So now you have to do data mining of the literature as well. That is also important. The reason is, again, you don't want to miss any targets, As I said, recently some target has been identified and whatever the database you have used that are not updated recently, so you will miss out that target. To avoid this, we are doing the data mining of the literature as well. So to do the data mining of the literature, I hope many of you knows how to do the SLR. So we are doing the SLR, systematic literature review. And by doing the systematic literature review, we will get to know if there is any targets which is not identified by the web servers, we include in the Excel file. Then our Excel file will become comprehensive. We need two comprehensive Excel file. One, the targets which your compound can hit. Second one is the targets which are involved in particular disease. So we are doing the data mining of the literature as well. And maybe many of you knows about mesh terms, Boolean operators. So we are applying these and we are, making a search strategies, particularly the persons from pharmacy practice or PharmD, they knows how to do the SLR and meta-analysis. So, if we want to know what are the target of routine in the drug resistance epilepsy, so we have to make the search strategies in such a way that so the only those articles will be reflected where rutin has been tested in the drug resistance epilepsy. So there is a boolean. These are the boolean operator, or, and. So by using or and, your complete search will be changed. If you are keeping or with the DRE or routine, so what will happen? You will get number of articles will be much more, because all those articles will be picked up where routine or DRE is mentioned. But your objective is not to see that kind of literature. Your objective is you want to see routine in DRE. So you have to be apply the boolean operator and instead of or, right? So you can do the in the PubMed. In PubMed, you click on the advanced options. In the advanced options, you can make the search strategies, and then you can click. Then you can do the screening of the literature. And to do the screening of literature, first you are doing on the basis of title, and then you can click on the save. You can create the abstract file. You can put the filter from here as well. If you want to see only the RCTs, so you can click on the RCTs. If you want to see all the articles, you can keep it unticked. And finally, whatever the articles you feel these are important, you tick select those boxes. And when you click on save options, and here you will select the abstract option. So what will happen by these options? Automatically, the article which you kept unselected, that will be excluded. Only the articles which you have selected in these boxes based on the abstract, only their abstract will be created automatically. So, we have a now various tools to speed up the literature process. Now there is a AI is playing a important role. So I request every one of you to use these AI based tool, which will help you to do the literature in a comprehensive way. So, in every project, literature is very important. Literature is playing a, important role. So to get the, any research gap or anything, you should have a comprehensive literature. So you can analyze, there is Elicit is the one of the tool where you can analyze the research paper at superhuman speed. So you can explore it, you can sign up and use it. There is a Lit Suggest is another one where you can, so you can get the similar studies based on your research interest. So it will give you the studies. If you suppose, for example, you are working on Alzheimer's and you want to see this particular molecule. So similar studies you will get by using these Lit Suggest. It will tell you by using the machine learning. There is a Litmap is another very good tool where you just add the, so if you are finding this one article is important for my research, just add the title of that articles. It will create the map of the other literature similar to that which is available in the literature. Got it? So you can understand, sometimes students comes to you and, so you are asking them, have you done your complete literature search? And students are saying confidently nowadays, yes, sir, I have done all the literature search on this topic and I have found only two articles. So, this is a challenging for the faculty to cross check whether there is only two article or more than that. So these tool will help you where you will just add the title of the one articles and it will create the lit map around that. So you can ask the student how you have missed this third article, how you have missed this fourth article. That is not in your results, right? So like that, you will get this lit map and you can get to know that what are the studies which are available in the literature with respect to that particular research, particularly where you are working, right? Then there is a Rian. I'm not going in detail. There is another tool, Rian, which we are using in our lab for the systematic review and meta-analysis. We are doing that part as well. So we are using the Rian. Rian is another good tool where you can do the blinding of the students as well. And then you can use it. You can do the screening of the literature in a more effective way. And you can come up with, evidence. There is a Distill R literature review software. This is a commercial one. If you have a money, you can purchase it. It will help you to do the systematic literatures. This one is Covidence is also, a commercial one. If you have a money, you can go with the Covidence. There is a Laser AI is a systematic review made faster, so you can use it. Now, step three, as I said, after getting the literature or all these things, you have to identify the common targets. So common target, you have to use the Venny tool. This is the web page of Venny tool, very easy to use. In one side, you add the targets which your compound can hit. Another side, you add the target which are involved in disease. This will give you the common target. So you can see that one side I have added the drug target. Another side I have added the target which are involved in disease. You can see that these 35 common I got results in between these two. So when you can use where you want to see the overlap, not only for the network pharmacology, for anything. if there are two things in your Excel file and you want to see how many are common in both these, so you can use the Vany tool for any purpose, for anything. Then there is a step 4, interaction among targets. String is the most popular one. So you can use the string cutoff value. We are keeping more than 0.7. This is the web page of string. So where you can, the next step is whatever the common target you are getting from this one, Vany tool, you are copy these targets and adding in the string database. So you can get the interactions among the targets by the string. So you can select the Homo sapien depending upon your research, and you can use the string. You will get a file like this, but you just copy it and paste in the Excel file. Your It will be arranged, and little bit manual work in the Excels you can do it. And one problems network pharmacological scientists are facing is they are saying that when we are creating the network, I am applying it for example, Alzheimer disease, and in my network there is a cancer signaling pathway is coming. So cancer signaling pathway is coming because you have not done the comprehensive Excel work. So in Excel work, you have to identify only those targets which have their role in that particular disease. So by disease targets, you will get lot of targets, which maybe some are important, some are not important. So you have to do that sorting based on your knowledge. So there is a biogrid, another one where you can check the interactions among the targets, but you can use either string biogrid or DIP is another one. Intact is another one, intact molecular interaction database. And then you can apply the VLOOKUP formulas and you can create the network. Creation of networks, you popular tool is Cytoscape. So just type in a Google Cytoscape, you can install it and you have to import the files and you can create the network in a different way. You can create in a circle way, you can create in another ways. So I am just skipping that part due to lack of time. So then after that, there is a CAG pathway. CAG is, Kyoto's Encyclopedia of Gene and Genome. So it will tell you about the pathways which are involved. So there is enrichment analysis. There is a, SR plot. So in SR plot, you can select the enrichment bubble. So by selecting the enrichment bubbles, you can get the informations about the pathways. Because up to that, you just get the informations about the targets. So these targets are involved in the some pathways, right? So SinyGeo is another tools where you can do the enrichment analysis, where you can get to know about the targets. So next step is after network pharmacology, you might have seen many research paper where they are combining network pharmacology with the molecular docking and molecular dynamics. So why that is required? That is required because by network pharmacology, you got to know that your compound can produce particular actions by modulating these targets. But how your molecule is interacting in the active site of the target, you don't know about it. So unless and until you are not doing the docking study. So docking study is particularly telling us about energetically favored binding conformations of the compound in the active site of the target. So to do that, you can, I think everyone of you knows about docking nowadays. Everyone is familiar with the docking. So docking is a, simple procedure. You have to be target structures. You have to be do the grid generations and all these. We are doing in our lab by using the Mastros of the Schrodinger target selection you can do from the protein data bank. There is a criteria for the selection of the target, because if you are adding in the search items acetylcholinesterase, you will get multiple PDB for it. So now question arise which one you have to select. So you have to select on the based on resolution value, mutations. You have to select based on the internal ligands and so on. So you have to prepare the ligand, target preparations, grid generations, and then you can do the docking studies. Then we are doing the MMGBSA calculations. Then we are coupling it with the molecular dynamics, where we are checking the stability of the molecule in the active site of the ligand. Molecular dynamics is depend upon the Newton's second law, that is the force is equal to mass into acceleration, and acceleration is changes in the velocity with respect to time. So these are the steps which you can do by using the GROMAC. You have to prepare the molecule, all these steps you have to do, and then we are calculating RMSD, that is the root mean square deviations. These are the screenshot of the molecular dynamics by using the Desmond module of the Schrodinger. This is the Desmond one. So Desmond is easy to use as compared to GROMAC. To run the GROMAC is freely available, but you should have a Linux for it. So these all parameters we are calculating: RMSD, RMSF, radius of gyrations, potential energies, free energy landscape, and principal component analysis. That is a PCA. So after that, you can combine it with the, if you have a facilities for the experimental. So if you are combining it with the experimental, so you can explore that by using the Western blot analysis or, RT-PCRs. You can do the cell cultures. So all these studies you can verify with. So what work we have done so far? This is just the last part. I think I have already completed the time. So these are the work which we have done recently in our lab. We have identified the repurposing of the drugs against hepatic encephalopathy. So we have used pharmacovigilance based approach, and then we have integrated it with the network pharmacology and experimental approaches. So we have identified, repurposing candidate by using the pharmacovigilance data, which is available in the FAIRS database. Then we have, identified the novel signals, means novel molecules which are not earlier repurposed. And then we have used the network pharmacology, molecular docking, and then we have done the experimental animal studies where we have explored the mechanism of, explored the effect of that particular drugs. And this the particular pathway we have explored with respect to repurposing of the drugs. We have identified carbidiol and amlodipine for the hepatic encephalopathy. This is another studies which recently our lab has done. We have selected natural compound against the drug resistance epilepsy. We have applied the network pharmacology, and we have tested luteolin and silymarin in the drug resistance epilepsy. So these are the major findings. And we have done the computational experimental animal studies, as well as the we have measured the neurotransmitters levels as well. Then this is the another studies where we have explored the mechanism of selected phytoconstituent as a galactose using network pharmacology and experimental approaches. We have used phenyl, strawberry, and fenugreek, and we have applied these tool. This is the another studies which we have done where these giloy, how giloy can enhance the immune response in mice immunized with JEB vaccine. So we have used the network pharmacologies with the experimental approaches, and this study has been published in the Phytomedicine Journal. This is another studies where we are using, as I said, for the pharmacovigilance, where we are identifying the novel signals and using. And these studies has been published in various journals. This is challenges with respect to network pharmacology is you have to be very careful about the data. Whatever the data you are using, that's why I am saying whenever you are using the data from the web server, read about the web server, who has created it, what are the limitation of that web server, what are the strength of that web server, then you can use it in a proper way. Quality of data, you have to cross check multiple data sources we have. So now you will means our team is working on a international to come up with a network pharmacological guidance document. Soon it will be published. It's under revision. So if you go through the papers with respect to network pharmacology, you will find that every researcher are using their methodology in their own way. So it should be standardized. So if someone of you is, if anyone is planning to work on network pharmacology, so there should be a standardized steps. This is the first step, this is the second, this is the third. So like that, it should be there. But when you go through the literature, you will get a lot of confusions. Someone is adding, means using some tool, someone is using some steps earlier and then later, and so on. So that's why, standard guidelines are important. So we are working in that area. So as I said, I hope next 2-3 months it will be come out. So conclusion is it's a next generation medicine research mode, and upcoming year you will find a lot of studies from India as well by using the network pharmacology. But of course, more in-depth analysis is required, and as I said, nothing can be replaced by the basic things. So basic knowledge is very important with respect to intracellular signaling pathway. Sometimes we have, if computer is showing that this is a first rank target, but by your knowledge, you know that even though it's a first rank, my computer is showing it's a first rank target, but I am not going with that. The reason is because I know if I am targeting this target, it will result in various adverse drug reactions. So if you have a reason, your computer is showing it's a first rank target, but you are with your knowledge, you are defending that I am not going with this target because reason is this, because it could result in various problems. So I will go with the second rank or third rank. So that can be happens. Acknowledgement. So I would like to acknowledge DST Serve, where I got two project, DST Serve SRG and CRG, ICMR. Three projects are ongoing. Sun Pharma, where I have worked, IIT Delhi, where I have done the projects works, Naiper Libraries, BITS Misra, THIST, ISF Moga, and my current organizations, of course, DIPSRU, and all my students and team members who are working hard to do such kind of studies. And again, I would like to thank this particular School of Pharmaceutical Sciences in association with Vijay Institute for such a wonderful event. If you have any queries, you can write me on this mail ID, anoopdpsru@dpsru.edu.in. So thank you so much for your patience. If you have any questions,
Yes,
you
sir,
can.
will do.
Yeah.
Thank you, sir, for your wonderful talk on network pharmacology. So you have made us aware about various tools and databases for the identification of target compounds as well as targets involved in disease, like Swiss, C, gene cards, stitch, etc. They are very much useful, sir. And this network pharmacology is a upcoming topic, and we see that you did a lot of research and published papers, and you were also focusing or stressing on how to analyze research papers quickly by using various tools like lit suggest, lit maps. Really interesting, sir. We have made us aware or familiar with these tools. Thank you, sir. And may I ask you one question, sir? I think
Yes.
your institution got a subscription of various software tools. Maybe if we want to work with you, or if we want to approach you in our work, is it possible, sir, with
Yes,
your
yes, ma'am.
institution?
Definitely, ma'am. That can be,
Uh,
we can work it. There's no issue.
we all can approach you with getting
Yes,
work
yes,
to
yes.
your softwares.
Yeah,
Yes,
sure,
sir.
ma'am.
Thank you, sir.
There's no issue.
Mm.
Thank
Very
you so much.
interesting session, sir. Thank you.
Thank you so
Thank
much,
you, sir.
ma'am. And thank you, everyone. Thank you. Thank you so much. Thank you.
So the afternoon session will be commenced from 2 o'clock onwards. I request all the faculty to be in the LH1 by 1:30. Thank you all.
Congratulations.